All right, in this set of slides, we're going to be looking at organic macromolecules. These are the molecules of life, the things that make up living organisms, make up cells. And we have 4 classes of them. We're going to be talking about the first 2 today, and then carbon as well. But looking specifically at what they are and the roles that they play. All right? Ultimately, what I'm going to expect for you in here is that you're going to be able to recognize each kind of these kinds of molecules, be able to say what it is that they do and why they're important, and also, how they work together. So we'll start here with the periodic table. Of these 94 naturally occurring elements, only about 26 actually play some sort of role in living systems. And of those, CHONPS, CHONPS, are the ones that are we're mostly interested in when it comes to macromolecules. All right. It's kind of interesting, actually, if you look at them all here. They're all sort of next to each other. We have this first row, we have hydrogen, and then the CH, or C-O-N and O, and then the phosphorus and sulfur. Now, CHONPS, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These are what make up macromolecules primarily. All of them have a carbon backbone. So all macromolecules have this set of linked carbon molecules. Now, they're often connected to things like hydrogen or otherwise, but one of the interesting things about carbon is that despite it being a fairly small molecule, it will make 4 bonds with other atoms, right? Other elements. And so what that means is carbon is very versatile when it comes to building large and complex structures, which are what we get with a lot of the macromolecules, especially when we start getting into proteins. Because protein structure is one of the most important things, the 3D shape. And having these 4-way connectors here with the carbons actually give us a lot of leeway when it comes to building shapes. Now, in addition to carbon, we also have hydrogen. All right, and actually, the way that we define organic molecules is not in how they were grown without pesticides or something like that, like we think about with the grocery store. Instead, an organic molecule is a molecule that has a CH bond. A CH bond is actually not a very strong bond, and because of this, it has a lot of potential energy. We don't tend to see a lot of these things, a lot of CH bonds outside of biological systems. It takes some energy to make these, right? And of course, living systems, one of their, hallmarks is the fact that they use energy. And so because of this, the C and H, these are fairly high energy bonds, right? So the more of these that a molecule has, the more potential energy that it is carrying. All right, let's talk about carbon a little bit. So carbon atoms make up, like I said, 4 connections with other atoms. This means that when it comes down to the 3D shape, carbon is actually fairly interesting. We often draw it just as like a cross and this sort of thing. But actually, in 3 dimensions, it's more of a tetrahedron, right? And the angle between, the bond angles between all these is about 109 degrees, 109.5 degrees. What it means is basically that these bonds are kind of pushing away from each other, and so you get this tetrahedral shape. Now, when it comes to carbon, carbon can form either single bonds, double bonds, or even triple bonds. As far as we know, it doesn't really form quadruple bonds. It only forms up to triple bonds. You can imagine another tetrahedron here being lining up so that it lined up here and you could get a triple bond, but in order to get a quad, you'd have to move this all the way down. That's just not happening. Right? So typically when we draw these things out, we have carbons right here, hydrogens here. You can see this is showing some hydrogens going back, some of them going forward, trying to show you some of that 3D shape. That's something that you'll do in organic chemistry, but it's one of the ways that you can show it that way. But these all have single bonds. Then we have here where we have a double bond, where we have the 2 bonds here between the carbons, and then we have ethyne, which are the triple bonds. Okay? So single, double, or triple bonds. We're mostly going to be talking about single and doubles. Now, we can have linear carbon, like I've shown you here, where we've got, let's see here, 1, 2, 3, 4, that's a hexane, right? Got 6 carbons together. Or you can start putting those into rings, where we take and connect the carbon not to another hydrogen and hydrogen here, but rather to itself. Here we have cyclohexane instead of just hexane. So we lose a couple hydrogens, but we get a 3D shape, or this kind of ring shape. In biology, we tend to see a lot of ring shapes for whatever reason. We can also get it where we can alternate double bonds here, and you end up getting a situation with just single hydrogen sticking off, and it's called an aromatic. Basically, all 6 of these kind of share electrons together, and it creates a particular kind of molecule. You can even throw a nitrogen in there too if you want. All right. So that's the 2 ways we really see carbons, in the linear or in these rings like this. Now, one interesting thing about carbon is because it has the capacity to make all these links, we can actually have carbon molecules that have the same chemical formula, but actually have different shapes. And there's 3 different ways that we do this. We call these isomers, right? So we have structural isomers, where we have right here, this is a butane and isobutane. If you look, C4H10 in both cases. But for this one, they're all on the line. This one right here, we've taken this carbon right here and moved it to this center one, and now we have it kind of as a cross, right? So a structural isomer is something where you move the carbon atoms around, and you are able to make a slightly different shape. So you get a different 3D shape, even though it has the same chemical composition. Then we have geometric isomers. So geometric isomers require some kind of double bonding. So normally a carbon here can kind of spin around this bond with another carbon, or with a hydrogen, or with whatever. But once you take and you put a double bond, like here, then you actually lock it into place. So it no longer is going to be spinning because it has those 2 bonds. All right? So when you have a geometric isomer like this with a double bond, well, you can take and have different, let's say you put a functional group on here. In this case, it's a methane, or a methyl group. You can either have those on the same side, or you can have them on opposite sides. So they can be cis or trans, all right? They both have the same chemical formula here, but this is a very different shape than this one is. This one tends to be more linear. This one will have more of a crick in it, right? And this becomes interesting when we look at things like fatty acids. Here we have a trans acid called allylic acid. Right here, this is the trans isomer. So if we look back to here, you can imagine that we had one part of the fatty acid going this way and the other part going this way. And you can see it's kind of linear, like so, compared to this one where it goes up and then kind of comes back down and has the kink, like we have here in allylic acid. All right, so these are geometric isomers. And trans fatty acids actually are produced, it's a industrial process where we bombard saturated fatty acids, usually from plants, with hydrogens, and we end up changing it into trans acids like this. These, of course, are very bad for us. But because they are linear, they tend to stack up well together and are solids at room temperature, so we can have things like margarine. All right, the last type of isomer are enantiomers. So these are situations where you have a carbon and it is bonded to 4 different functional groups, right? And when you do that, there are 2 ways you can do it. You can have a mirror image of this, all right? It only happens when you have 4 bonds and it's to 4 different things. But when you do that, if you look here, if you slice this right down the middle, this one is the mirror image of this one. So where this one might fit, this one will not necessarily fit. All right? This means that you can have things that are almost exactly the same, except for their shape is different enough that they have different functions. Example of this is ibuprofen. Ibuprofen, one of its enantiomers is a pain reliever, the one that we use. The other one does nothing. Thalidomide was another example of this. This is probably not something you've heard of so much, but back in the 70s, they started giving this to women who were pregnant women to help with morning sickness. And one of the enantiomers actually does a really good job of helping with morning sickness. It's really effective. The problem is the other enantiomer actually causes birth defects, and they can switch back and forth with each other. And so they found out fairly quickly that thalidomide was a bad idea. And because of the other enantiomer was causing birth defects. All right, so with that in mind, last thing I want to talk about with carbon is some of the common functional groups that we tend to find on these. And there are 5 of them that you are going to see a fair amount of, and so you should be familiar with them. So these are just common combinations of atoms that can be attached to the carbon chain. So imagine R here is your carbon chain, right? R basically suggests that there's something over here, all right? And then this is the thing that's stuck on. So we have a couple different possibilities. One is a hydroxyl, so it's just an OH. It's stuck on there. You add this on and it actually makes the molecule polar. It adds some polarity because the oxygen is pulling electrons towards itself. Get a partial positive, get a partial negative. A methyl group is a little bit different. This is mostly nonpolar. So you put one of these on here and you get a hydrophobic piece on that's bonded on the carbon. All right? Methyl group is also very common. A carboxyl group. We have a carbon with a double bonded oxygen and this OH here. When you have this situation, it tends to pull the electrons enough from the H that the H actually usually is dropped off. When that happens, it's an acid, right? So it contributes a hydrogen to the solution, and so carboxyl groups often make acids. Amino groups with this a nitrogen and 2 hydrogens here. Often you'll get a H plus that will join on here, and you actually end up getting actually more. So they end up pulling some H plus from the solution and end up actually becoming basic. All right, slightly basic, right? But an amino group, you'll see this. You've heard of an amino acid. All right, well, it has an amino group on it. And then lastly, we have our phosphate. So it's a P with 4 oxygens stuck to it. Remember, it's the road down, so it can make more connections. But these will also release H plus, which means that it also becomes acidic. Charged, this is actually a charged molecule right here. Amino group can be charged as well. Then we got our acids here, and then the hydroxyl and the methyl. Be familiar with those, and you should be able to recognize them. You will see them a lot. All right, let's get into our actual molecules. So in general, our macromolecules are giant molecules that are made of small pieces called monomers. So the big molecules are polymers, which means that they are made up of lots of small pieces called monomers. So you need to be familiar with each one of these monomers, all right? Lipids here, the only real monomer that we talk about with lipids are fatty acids. When they're polymer form, they'll actually be the polymers will actually be like triglycerides. So that's a little bit off on how that's saying there, all right? So the monomers are the fatty acids like this. Carbohydrates are monosaccharides. Proteins are amino acids. And nucleic acids are the nucleotides. So in order to make a macromolecule, you don't make it from scratch necessarily. You take these that are already made, and then you put them all together to make the larger molecule. So let's start with our lipids. Now, our lipids are kind of our weird ones. They have probably the most diversity in their structure. They tend to be large, mostly hydrocarbon, and they are mostly hydrophobic. In fact, that's the one thing that ties them all together. This right here and this right here are very different molecules, but they are both hydrophobic. And so we put things like steroids here and fatty acids into the same category because they are both hydrophobic. Okay? All right. Let's see. Do we talk about saturated fatty acids? I'm going to talk about saturated and unsaturated fatty acids right now. So we'll talk about steroids in just a minute. Saturated fatty acids, you have this long hydrocarbon chain. You got just a few oxygens on one end. That helps it connect to other things, but it's not enough to actually make it dissolvable in water. We have 2 kinds of fatty acids. We can have the ones here that are saturated with hydrogens. If you look, each carbon is either connected to 2 hydrogens or to 3, right? It is the maximum number of hydrogens we can put in there, so we say it is saturated. This one has a double bond. So not only does that mean that you can form a geometric isomer, but you can also, you also make kind of a kink in the chain, or you can make a kink in the chain. So you take that double bond in there, you take away a hydrogen here and a hydrogen there to make that bond, and it is now unsaturated. Because these tend to form more kinky sort of chains in the 3D shape, we tend to see unsaturated fatty acids in things like oils. They are not, they don't stick together well enough to be solid at room temperature. We tend to find our saturated fatty acids in things like butter, which its lovely self, sticks together pretty well, and is usually at room temperature is a solid. All right, so saturated fatty acids tend to be solids at room temperature. Unsaturated, because they don't stick together as well, tend to be liquid. All right, they're going to talk about four different lipids. First, we have our triglycerides. Tri, meaning three. We have three fatty acids stuck together with a glycerol molecule. All right, this is a very highly energy, it has a ton of energy in here. If you look at all of the potential energy from all those CH bonds, that's something you see here in all of these, right? Is a lot of potential energy from the CH bonds. You take a bunch of these, you put them on a glycerol molecule, and now you have a triglyceride. These are the fats that we are most commonly interact with: oils, butter, things like that. Their function is long-term energy storage, right? So they are the most energy efficient because they have so many carbon-hydrogen bonds. They also act like padding. You can see the big fat bear here, right? In the stream, it's getting ready for hibernation, and so has gained a lot of fats for energy storage over the winter, insulation in the cold weather, and padding for, sleeping on your side for 7 months. All right? So we have our glycerol, we have our fatty acids together. It's a triglyceride. Next kind, phospholipids. Phospholipids are interesting because we have is a glycerol molecule connected to two fatty acids, kind of like we saw here. Except for the third one, we exchange it for a phosphate. Well, that phosphate then changes the whole character of this thing, because now we no longer have a totally hydrophobic thing, but we have a partially hydrophilic section. This part right here gets along well with water, while this one doesn't. So when you take these things and you dump them into some water, the hydrophobic tails tend to want to stick together, and the heads want to be out by the water. And so what you end up with is a two-layer membrane like this. So they form a bilayer membrane here with a hydrophobic section in the center and then the hydrophilic portions on the top and bottom. This makes a very effective barrier now. Anything that is dissolvable in water on either side of these is not going to go very well through our hydrophobic center. All right? So we use phospholipids to form barriers, things like cell membranes, membranes inside of cells. These do a good job, and actually even milk. Like milk fat globules are actually surrounded by phospholipids as well. That's what allows them, fat in milk, to actually dissolve in the water. Right? So compartmentalization of cells, the ability to, form a barrier, have a membrane. That is because of the phospholipids and their hydrophilic and hydrophobic characters. All right, waxes. Waxes, their primary thing. So what we have are two phospholipids, not phospholipids, two fatty acids that are connected together with something called an ester linkage. It's just two fatty acids connected to each other, a whole bunch of nonpolar, right? They stick together, we end up getting coatings which either keep water in or keep it out. So we have something here like a beehive or on this leaf. They're storing things like nectar in here and honey. And this is, because wax is hydrophobic, it does a pretty good job of keeping the water in, all right? Or in this case, keeping water out. They are hydrophobic coating, basically allows us to conserve water, especially in plants. It's a big thing in plants, but we do see it in other places as well. And then lastly, let's talk about our steroids. So steroids all come from, a kind of basic cholesterol molecule. All right, it's this 4-carbon or 4 aromatic. Well, they're not all aromatic, but basically 4 rings. All right, 4 carbon rings, and then we add some stuff on either side. This is the cholesterol right here. We find this in cell membranes. From cholesterol, we can actually make estrogen and testosterone. If you look at these things, it's remarkable how similar they are, right? Considering like how different the effects are of testosterone and estradiol, so estrogens. I mean, we're mainly talking about an OH here versus a double bonded O, right? So we lose some of these double bonds here, but that's really the only difference, which is kind of crazy. Now, these are also hydrophobic. So they don't dissolve in water, but they still play a major signaling role. We find them going through the blood. But in order to do that, they have to be bound to carrier proteins, right? So these are some of our most potent hormones, but they are always in concert with these carrier proteins so that they can actually go through the blood and carry those signals. All right, so that's our 4: triglycerides, phospholipids, waxes, and steroids when it comes to lipids. Now let's do the carbohydrates. So carbohydrates. We usually talk about 2 different kinds of carbohydrates. We have our monosaccharides. These are monomers, but the monomers actually have their own function as well, all right? But we can take and put those together into a larger molecules called polysaccharides. The 2 of them have slightly different functions, but this is one of the cases where we have a fairly important function for one of the monomers. All right, now typically we see our carbohydrates in, it's carbon, hydrogen, and oxygen primarily. Occasionally you get some other things in there, but the primary thing is a carbon, hydrogen, oxygen. Usually if you have anything else that's on there, it's kind of stuck to the outside, right? And it's in a 1 to 2 to 1 ratio. So when we come to here from our fatty acids, right, we're basically exchanging a lot of hydroxyl groups for hydrogens, right? We're getting rid of a lot of those hydrogens and we're adding hydroxyl. So we're taking away energy potential, but we're adding a lot of polarity to this molecule. And so it ends up being something that, unlike fats, dissolves really well in water. We typically see, this is a glucose molecule in a linear shape, but we typically see these things. You'll connect the OH here with the O right there, and you end up with a ring shape, right? And for whatever reason, we tend to see the ring shapes like this. Now you can put your you can have your H on either one side or the other. And if you look, these are actually geometric isomers of each other. Because even though there's not a direct double bond, there's 2 bonds right here on this carbon. It can't spin, so we have alpha glucose and beta glucose. All right, the function of these is for energy, right? Every other kind of thing that we use for energy, it all goes through monosaccharides before it can be made into ATP. ATP is the thing that we ultimately use in the cell for energy. In order to get to ATP, we have to go through some kind of monosaccharide first. All right, so that is that's the primary role of monosaccharides, is to be broken down for energy. Now, polysaccharides do a lot of different things. We can do energy storage. They're less efficient than fats because they have a lot of these OHs here, but they are often used for that, structure, and then also recognition between cells. So in order to go from monosaccharides to polysaccharides, we go through a process called dehydration synthesis. Dehydration synthesis takes 2 different monomers here, extracts a water. What you do is you take, see this OH here and OH here, right? If you take water out of, you say 2 H's and 1 O, that leaves you with just 1 O, right? You have an enzyme that does this. It's a fructose 6-phosphate synthase, in this case for making sucrose, a chitin synthase, in this case for making chitin. But you're taking the 2 monomers and you're trying to stick them together into a polymer. You take and you end up with this O here bonded to 2 carbons. It's not showing the carbons, but usually in these situations, the nodes here are carbons. All right, so you have your 2 carbons that are bonded together with an oxygen in the middle plus water. Because we are taking water out, we are dehydrating it, and we are synthesizing because we are putting things together. So that is what dehydration synthesis is. And fortunately, all of our macromolecules get put together using dehydration synthesis. So you only have to remember one kind of reaction to do this. Now, we have the name for these kinds of bonds here. So we have different names for the bonds. So sorry about that, but we do have different names for the bond. The bonds are called glycosidic bonds. So that's our synthesis. If we want to break them apart, we just go the opposite way. We take water and stuff it back in to this bond, and it splits. So we take water, which is hydro, and we lyse it. Hydrolysis. This is taking water and splitting the bond and getting your monomers again. All right? So dehydration synthesis puts them together. We put them together, we pull water out. Hydrolysis, we use that water, we put it back in again, and we get our monomers. The bonds that we are making are called glycosidic. And you see this glyco right here? Anytime you see that, that's a pretty good sign that there's a sugar involved. Okay, so that is how we take and break those bonds, is hydrolysis. We make them with dehydration synthesis, break them with hydrolysis. All right, polysaccharides. So what are some important polysaccharides? Well, like I said, there are two main functions. We have structure and we have energy storage. The structure is mostly in cell walls, all right? So we have cellulose cell walls in plant cells, chitin in fungi cell walls, and peptidoglycan is in bacteria, all right? So anything that has a cell wall, it's going to be made of partially, at least, of polysaccharides. So that's one of the big functions of them. Things like wood, it's all sugar, right? It's all put together in, glucose, all put together in these long strands here, but that is what it is. It's a polysaccharide. Now, starch and glycogen are for energy storage. Starch is what plants use. We can break this one down. Same thing with glycogen. We can actually use this. Starch, we tend to see it in things like seeds and potatoes, any place where plants need to store energy, all right? So that's in starches. Our glycogen, we store that in our muscles and our liver. This is something we can fairly easily clip off these sugars to use for energy. It is pretty quick energy storage. However, if you look, these do not store as much energy per unit weight as our fats do. However, they're easier to get to. And so plants, which I guess don't need aren't as worried about things like weight, just use polysaccharides for most of their energy storage. We humans, we animals, tend to use things like glycogen and then fats for our long-term energy storage. All right, one last thing. Polysaccharides are important for recognition. On the outside of the membrane, you have polysaccharides sticking out here, fairly short usually, but these things are recognized by the immune system. This is one of the ways the body knows that a cell is either supposed to be there or not, right? If it's your cell, then it will have the right polysaccharides on there. Your immune system will recognize it and it won't attack it. If it doesn't have the right ones on there, it's going to attack it, right? A lot of times that means that there's a pathogen, some kind of invader that has gotten into you and your immune system needs to kill it. Other times, maybe you got an organ transplant, right? And so if you've ever heard of people taking anti-rejection drugs, what they are doing is making up for the fact that they don't have the right carbohydrates on the outside of the cells. And they have to knock down the immune system to be able to do that. It's immunosuppressant, and so that it doesn't start attacking cells on the organ, right? But ultimately, it's these polysaccharides here that help identify the cell as being self. It belongs here, all right? So that's the last role that we have of our polysaccharides. And that's that. Thanks for sticking with me. I know it was long.